All right, we're good to go. All right, this lecture is vestibular function 1. And here's our outline for vestibular function 1. And I'm going to divide it into 3 videos. So in the first video, I'm going to give you an overview of vestibular function, what it's important for, what it does. Then we're going to talk about labyrinths. There's 2 types of labyrinths we're going to talk about, the bony and the membranous labyrinth. And then we're going to talk about hair cells, which are the sensory cells. So we'll go through transduction, the structure of these hair cells, and how they communicate with the nervous system. So that's going to be the first video, which will be video A. In the second video, which will be video B, I will focus in on semicircular canals. The hair cells in these semicircular ducts or canals detect angular acceleration, and we'll go through how those do that. And then in the final video, which is going to be video C, I will talk about the otolith organs and how they detect linear acceleration, so acceleration either vertically or horizontally. And then after that, we're just going to briefly look at the circuits, and that'll be kind of an introduction to vestibular system 2, which we're going to talk about vestibular reflexes. So that's our course for this lecture. So let's switch back over to the slides. So our vestibular system is kind of interesting because most people aren't aware of it unless something goes wrong. So the vestibular system has a couple of main functions that are important. It's very important for balance and coordination. So these are functions. And it turns out that this first function, the vestibular system, works very closely with our proprioceptors and our visual system. So we'll put that under here too. Works with proprioception and vision. So all 3 of those are going to work together to help us with balance. The vestibular system, another function of it, is orientation of our body and head with respect to gravity, or detecting the orientation of our head. So of the head, WRT is just with respect to gravity, so that we know where our head is in space, and sometimes that's going to result in a correction. So it's going to be orienting the head, and the body sometimes too is also involved. And then it also is very important for coordination of head and eye movements. So those are our main functions of the vestibular system. Like I said, these are all things we kind of do automatically, so we don't usually pay attention to them unless something goes wrong. So what can go wrong? One thing is something like motion sickness, and then we get a mismatch perhaps between our visual system and our vestibular system, and then that's going to make us feel nauseous and kind of dizzy because those 3 systems aren't bringing in the same information. So we're going to start talking about the peripheral apparatus first, and then that can help us understand how the vestibular system is able to pick up this sensory information. So our vestibular system is located deep in the head, so it's near the ear. As a matter of fact, the bony labyrinth that surrounds the vestibular system also surrounds the cochlea, which processes and brings in auditory information. So our vestibular system is composed of semicircular canals. There's 3 sets, so there's 3 on each side of the head. There's a horizontal, a posterior, and an anterior semicircular canal. These detect angular acceleration. Then we have, located in our vestibule, we have what are called otolith organs. And those are going to be responsible for detecting linear acceleration. So this bone that surrounds these membranes that contain fluid. This is our bony labyrinth, and it's deep in the temporal bone, which is the hardest bone in our body. And then suspended within that bony labyrinth is our membranous labyrinth. And that's what makes up these semicircular ducts as well as our vestibule here. And then, like I said, they're actually connected to the cochlea, which is part of the auditory system. So we're going to take this and look at it in a diagram form so I can talk about some other characteristics of the labyrinth. So in this diagram here, this black part on the outside here is going to represent our bony labyrinth. And then this purple with the green splotches in it is going to be our membranous labyrinth. So the first thing we need to understand is that there's different fluids within each of these compartments. So within the bony labyrinth, we have something that's called perilymph. And perilymph is low in potassium and high in sodium. So that's going to be filling all these spaces between these dark black outlines. And then again, suspended in the bony labyrinth is our membranous labyrinth. And this has a different type of fluid. The fluid in here is called endolymph. And endolymph is high in potassium and low in sodium. And this difference between the endolymph and the perilymph is very important for the ability of this system to react quickly. Each of these, so here's our duct, and you might notice that the endolymph, all these semicircular ducts here, as well as the utricle and the saccule, which are our otolith organs, and the cochlear duct, they're all continuous. So we have endolymph that's all throughout this whole duct system, and it turns out that endolymph is produced continually and automatically, and then it gets absorbed through this endolymphatic sac. So that's similar to some other things we've talked about in that we have a closed system with continual production of fluid. So if there's a problem in that system, if we get a blockage so that that fluid cannot be reabsorbed, we get swelling of this duct, and that can affect our auditory system and our vestibular system. And that's called something called Meniere's disease. Let's put that over here. So Meniere's disease is a swelling, usually due to increased fluid, specifically endolymph. And because this endolymph goes throughout the vestibular and the auditory system, it leads to auditory problems. So this can be problems hearing, or it can be tinnitus, which is a ringing in the ear. And it also leads to dizziness and nausea. Okay, so let's talk about the receptor cells and the receptor patches. So within each of these membranous systems here, you can see these little green patches. These green patches are the sensory epithelium where the sensory hair cells are located. So notice that we have one at the base of each semicircular canal. So here's one semicircular canal or duct, and here's that patch of epithelium. Here's another one for the anterior semicircular duct, and one for the posterior semicircular duct. And then the utricle and the saccule also have their individual patches of sensory epithelium. So within these sensory epithelial areas are something called hair cells. And I'm going to draw those on the board, and we're going to talk about these hair cells in detail. So these are our receptor cells. I'm going to erase this. Board is hard to erase. Okay, so these are hair cells. And those are our sensory cells. And I'm going to draw a couple of them. So they look something like this. They have a nucleus. And they have something called a hair bundle at the apical part. So apical means top. And they have this hair bundle that has this staircase-like arrangement of these structures called stereocilia. So they make up the hair bundle. In the vestibular system, next to the tallest stereocilia is a structure called a kinocilium, and it has a little knob on the top. So this is a kinocilium. And these hair cells, as well as hair cells that are next to supporting cells, so I'm just going to draw kind of a random supporting cell here. That has a nucleus too. These are all connected by tight junctions. So you guys have heard of tight junctions before. Tight junctions help limit the flow of things between one side where the tight junction is and the other side. And that's what they're going to do here too. So it turns out that bathing the stereocilia, or the hair bundle of these hair cells, is that endolymph that we talked about. And remember, that's high in potassium. Okay, so that's our endolymph, high in potassium. And so that's going to be all around our hair bundle here. And then the basolateral part of the cell, so that just means the sides and the base, is going to be bathed by perilymph. Okay? And so then if we tie that back to our diagram over there, we would have our bony labyrinth somewhere out here, the bony part of it. Okay, so here's our perilymph all throughout here. So hair cells are mechanoreceptors, so they're going to respond to movement, and specifically movement of the fluid. I'm going to go to the other side. So when our endolymph is moving, those hair cells within the endolymph are going to be affected by that, and specifically the stereocilia hair bundle. So if we have a hair cell, which way did I draw this? Okay, I drew them that way. And we have the kinocilium here, and then our stereocilia that go this way. And we have movement towards the kinocilium, or towards the tallest stereocilia. What's that? That's going to depolarize this hair cell. Okay, so this is going to result in a receptor potential. Which you all know is graded, and that is going to increase the release of glutamate from the hair cell. And that glutamate is going to then bind to glutamate receptors. On the vestibular part of the vestibulocochlear nerve, which is cranial nerve 8. Okay? It turns out when our hair bundle is not moving towards either side, so these are just straight up and down, so we could think of this in the absence of stimuli. Our hair cell has baseline activity. So that means it's slightly depolarized at rest. and it's going to be releasing a little bit of glutamate. Okay, so we're going to draw a couple little dots in there. And again, it's going to go on and contact the glutamate receptors on cranial nerve 8. We can also have our hair bundle moving in a different direction. So that hair bundle could also move away from the kinocilium or towards the shortest stereocilia. And that would be in this direction. That happens to close mechanically activated cation channels. So that's going to result in a hyperpolarization of this hair cell relative to rest. And we're going to decrease the amount of glutamate that's released. And so we're going to decrease any excitatory effect on the associated primary afferent. Okay? So hopefully that makes sense. And as I told you a few minutes ago, we have different fluids bathing the apical part and the basolateral part of these cells. So in this case, when we move the hair bundle towards the kinocilium, towards the talus stereocilium, we're going to get an opening of cation channels. And that's an increased opening relative to rest. So it turns out that in the vestibular system, the charge carrier is actually potassium. So that's unusual. Okay, that's what's going to end up depolarizing the cell. So potassium is the charge carrier. And again, at rest, there's some of these cation channels open. And so there's going to be some potassium going in, but not as much if it's depolarized or if it's bending towards the talus stereocilium. And then when the hair bundle is bent towards the shortest stereocilium, there's going to be a closure of these cation channels. And that's going to result in hyperpolarization and a decrease of glutamate. So that's our process of transduction. So transduction, as you know, is the process of converting some kind of a external stimulus into neuronal signals by a receptor cell in a sensory system that can then be carried to more central aspects of the central nervous system. So that is that, and then I have a couple of slides about these hair cells. So this is a diagram from your book showing these hair cells, similar to what I drew on the board. So here we have those hair cells. There's the stereocilia, or the hair bundle at the top. We have endolymph bathing the apical parts here. We have tight junctions between the cells to separate that endolymph from the perilymph. And then we have these hair cells. They don't have axons, but they do release glutamate, and they release glutamate onto the vestibular part of cranial nerve 8. Now, one thing this slide does show you that I forgot to mention is that in the vestibular system, these hair bundles are embedded in this gelatinous matrix. So what color do I want to use? Maybe I'll use red. I have limited colors up here. So in the vestibular system, we have this gelatinous mass that the stereocilia or the hair bundle is embedded into, and that's shown on this slide here, and that's what the G stands for, is this gelatinous mass. This is a scanning electron micrograph of a hair bundle at the top. So this is the top of the cell. Here's our hair bundle. You can see that nice staircase arrangement of the stereocilia. This right here is the kinocilia, which has an arrow pointed to it, and that's found in the vestibular system. This over here, these arrows point to these little filamentous proteins that are between the stereocilia and the hair bundle. These are called tip links. And these tip link proteins are important for gating those cation channels open so that when you move the hair bundle towards the tallest stereocilium or the kinocilium, you're going to get pressure on this tip link so that it stretches. That's going to gate open these cation channels. And remember, it's potassium that's going to go in and depolarize that cell. So I'm going to stop there for this video. And then in the next video, we're going to talk specifically about the hair cells in the semicircular canals.